Hi, my name is Sahib, and I'm going to be presenting this paper. Exosomes derived from hepatocellular carcinoma cells induce sorafenib resistance in hepatocellular carcinoma, both in vivo and in vitro, for my biology of cancer presentation. Hepatocellular carcinoma is liver cancer, and according to the paper, it's the fifth most common cancer and second leading cause of cancer-related deaths worldwide. That's an important point to note. It's one of the deadliest cancers, I guess, because a second leading cause of cancer-related deaths worldwide. So it's important to investigate and learn about this cancer. I was interested in this paper though because of the mention of exosomes. Although I haven't previously studied exosomes in depth or anything like that, I was interested to see how exosomes played a role in, propagating cancer, or in this case, inducing drug resistance again, or inducing drug resistance in cells against cancer drugs. So yeah, that was pretty interesting to see, and especially in this case because the paper says that sorafenib is currently the only medication approved for the treatment of liver cancer. So anything, and in this case exosomes, something that would be inducing cells with the resistance against that drug would be highly detrimental to cancer treatments for this cancer. So it would be important to research this and try to find ways to, maybe inhibit exosomes or HCC, liver cancer exosomes, to, help in the sorafenib delivery, as they actually show in this paper. Although they do it in vitro, it'll be interesting to see. So basically, in the methods, as you can see, they got exosomes from two different invasive liver cancer cell lines, MHCC97L and MHCC97H. For simplification, I'll be referring to these as 97L and 97H. There's also another non-invasive, non-cancer cell line that they also got exosomes from that, so regular exosomes. So yeah, and as we go on, the introduction, it shows exosomes play important roles in exchange of biological information and substance transport carriers. So they're kind of like communication vesicles. And exosomes are vesicles found on the inside of cells. And they're also found in many blood fluids, as are present in many and perhaps all eukaryotic fluids, including blood and urine. They're investigated in many tests to identify diseases and whatnot. So basically, we'll be investigating that. And so the paper also says that exosomes induce sorafenib resistance through the HGF c-Met AKT signaling pathway. So we actually learned about this pathway in class, and it'll be interesting to see how they measure it. Oh, one other important point to mention, they started research on this paper because they observed in previous studies, suggested that uh, that exosomes played a role in drug resistance, and also they enhanced AKT and MAPK, or ERK signaling pathways, which enhance tumor cell invasion. So the first experiment, or the first assay we'll be looking at, is this one right here. So this is an apoptosis assay, which we learned about. And there are 5 different ones carried out, and they were carried out for samples of 10,000 cells. 10,000 cells were analyzed. So an apoptosis assay basically numbers the amount of cells that are in apoptosis, or in necrosis, or in living cells. So each one of these little dots right here is basically a cell. And depending on which quadrant it's in, as we learned in class, it's either necrotic, as shown in this quadrant, a late apoptotic, as shown as this in this quadrant, early apoptotic, as shown in this quadrant, and living cell, as noted in this quadrant. So how the assay does this is there are two markers. The first one is annexin 5, and annexin 5 is shown on the x-axis, and it increases as we go on to the right. So annexin 5 binds phosphatidylserine, which is a protein in cells which binds to annexin 5 when it comes out. So when phosphatidylserine is out, the cell is apoptotic, the annexin 5 binds, and the higher annexin 5 binding is shown on this graph. On the right, there's higher annexin 5 binding, so apoptotic cells are shown on the two right quadrants, right over here. And the other corresponds to or sorry, not phosphatidylserine, but propidium iodine binding, which binds to DNA, which is exposed when cells are apoptotic. So high propidium iodine binding as you go up, and as you can see here, late apoptotic cells, excess their DNA, so it's high over here. But although high propidium iodine binding, along with low annexin 5 binding, corresponds to necrotic cells, not apoptotic, which is shown in this quadrant. So the first assay we see here is basically the control. Regular cells, mostly living, nothing odd or nothing much to note about that, as they're seen in this quadrant. However, as we move to the sorafenib apoptotic assay, we can see sorafenib induces apoptosis in cells. So right here you can see a higher amount of apoptosis as compared to the control. But then we move to the next one, and this is sorafenib plus LO2 exosomes. So LO2 exosomes are regular exosomes that are exosomes derived from regular non-invasive non-cancer cells. So how does the addition of these exosomes lead to a difference? So we can see there's a slight decrease in apoptosis. Not too much to note, almost I think 3% in each quadrant of apoptosis. You see, it goes from 46 over here to almost 43, and then from 15 over here to 12 over here. So almost 3% decrease in apoptosis. So the exosomes might have helped that decrease in apoptosis, but we note going on to the next assay that sorafenib mixed with 97L exosomes. Now remember, these exosomes are the ones that are derived from one of the liver cancer cell lines. So these are highly invasive or highly invasive cancer exosomes, and they resulted in a much bigger decrease in apoptosis. And remember, sorafenib is still treated, so when we compare it to this assay right here, 46% here, but 36, that's a 10% decrease in apoptosis. And then over here too, although there was a there was not much of a decrease for the early apoptotic cells. There were less in that to begin with anyway, but we can still see there's a 10% difference in the late apoptotic cells. So these exosomes are clearly reducing the amount of apoptosis that sorafenib caused. So essentially, they're in, well, as the paper says, inducing resistance against sorafenib by lowering the apoptosis rate. And as we can see over here, as we go on to the next one, there's a bigger jump down. So this was 36%. Now this is only 25%. So this lowered the rate of apoptosis even more, or the amount of apoptosis even more that was induced by sorafenib. So, and it notes that over here too, that this cell line, the 97H liver cancer cell line exosomes, were even more aggressive in reducing the amount of apoptosis caused by sorafenib. So basically, they allowed for more of the cells to survive and go on. And in the context of cancer, this would be bad because we want apoptosis for cancer cells. So the 97L exosomes and the 97H exosomes, although 97H was much more effective in reducing the apoptosis rate and allowing cells to go on to grow. So that's what it basically shows over here, and you can see the apoptosis rate in a simple graph in this figure, figure B right here. And yeah, again, sorafenib plus 97H exosomes greatly reduces it when compared to just sorafenib. As we go on to the next figure right here, this figure examines the the HGF c-MET AKT pathway that was mentioned, HGF c-MET AKT pathway. So as the last figure showed apoptosis and how the exosomes reduce the apoptotic rate, inducing resistance against sorafenib. This is how they do it basically, by activating this HGF c-MET AKT pathway. Now, for this, there's a figure right here. So this pathway, basically, it starts with a c-MET right over here. So a c-MET is an HGF receptor, or an HGFR, and HGF is hepatocyte growth factor, and this is its receptor. So hepatocyte growth factor comes along, binds to MET, and this is a tyrosine receptor tyrosine kinase. The MET phosphorylates, and what happens is PI3 kinase comes along, binds to it with its P85 subunit, which causes it to become activated. So there's more activated PI3 kinase, which, as we go on to this figure, we can see that PI3 kinase results in a change of PIP2 to PIP3, which causes PDK to phosphorylate PKB, and PKB is AKT. So more phosphorylated AKT, which phosphorylates many more things, and it's a pro-growth pathway, this AKT pathway, as we learned in class also. So the exosomes basically activate this pathway, as we're going to see over here. So in the first graph right here, we can see HGF levels in the sample. So in the control, there's not much HGF. In the sorafenib, there's even lower HGF. But you can see in the 97L exosomes and the 97H exosomes, there's an increase in HGF. So that would mean this pathway, HGF c-MET AKT pathway, is being activated in the presence of the 97H and 97L exosomes. So yeah, and over here in this graph, especially in this Western blot, we can see that this is phosphorylated MET. So phosphorylated hepatocyte growth factor receptor, so the activated receptor, is increased when compared to sorafenib. So in this line right here, there's sorafenib inside the cell. So that's inhibiting the pathway. I guess, well, not the pathway, but there's the regular amount over here. But there's an increase when you add the 97H exosomes. So this pathway is being triggered even more in the presence of 97H exosomes. And in this one, you can see phosphorylated AKT is in a higher amount in the 97L and the 97H exosomes as compared to just sorafenib. So these two exosomes are activating this pathway. As you can see, there's more phosphorylated AKT, although it doesn't show much more for 97L exosomes, much more phosphorylated receptor, but it's basically there. But you can see it in the phosphorylated AKT. And there's also more phosphorylated VEGFR2 receptor, which plays a role in angiogenesis and spread of cancer through metastasis. So that's very interesting to see. So these two exosomes are the liver cancer exosomes are activating the pathway, which leads to growth, and that's how they're lowering the apoptosis rate and inducing resistance against sorafenib. So in this next one over here, this graph is showing sorafenib in conjunction with another drug, crizotinib. So crizotinib is a c-MET or a MET inhibitor basically. So when you add this drug, there's sorafenib in every one of these cells. When you add crizotinib along with the 97L and 97H exosome, you can see there's a decrease in phosphorylated MET. So this pathway is less activated now. So that means the crizotinib is helping inhibit this pathway, and helping the sorafenib to do its job basically. And you can see over here in the 97L exosome and 97H exosome, there's less phosphorylated AKT as well. And the same thing over here, but over here they have the MK2206 drug, which is, so the MK2206 is a phosphorylated AKT inhibitor. So what that does is there's less phosphorylated AKT, as you can see over here. And over here, that means the pathway is being inhibited, so growth is being inhibited, so the cancer cells would not be able to grow in conjunction with sorafenib and MK2206, as compared to when there's no MK2206, as you can see over here, much darker, more phosphorylated AKT, and much darker over here, more phosphorylated AKT. In this case, there was only sorafenib and the 97H exosomes, basically. So yeah, that shows how how exosomes induce resistance in cells via the HGF-c-Met AKT pathway.